to join.
Okay, ma'am.
In the last class, we were talking about cleavage and different patterns of cleavage. Are you able to see the screen? Able to see the screen?
Yes,
Yes,
ma'am.
ma'am.
Yeah. So we were talking about cleavage and what is the importance of cleavage and what are the different types of cleavage. And initially, the zygote converting into multiple cells, that is a morula, morula converting into blastula, blastula converting into gastrula stage, and the gastrula stage leads into the formation of organs. During this process of gastrula converting into the different organs like eye, nervous system, or limbs, or it is alimentary canal. During this process, there are certain aspects or the biochemical changes that are occurring that is called as fate maps formation and differentiation. So when we are talking about blastula converting into gastrula, we have seen different stages of gastrula, which is called as a coelogastrula or discogastrula. I'm just recollecting. Okay. And then we also have seen what are the different planes of the cleavage. And we have seen that there are vertical planes and the horizontal planes, equatorial planes or the latitudinal planes, or it is the meridional planes, which are dividing the zygote into different cells. And these cells are called as a blastomeres. And these blastomeres are arranged in a single order or a single line or single layer, having a space in between that is called as a blastocele. So this is what we have seen. And then coming to different types of a cleavage, we have already seen the different patterns of cleavage, that is a holoblastic cleavage and the meroblastic cleavage. In the holoblastic cleavage, we have seen the radial cleavage, where radially the cells are being divided right in the central axis. Then bilateral, we have seen the bilateral symmetry, wherein we have seen that both the sides are the mirror images of each other. First will be an meridional and the second will be perpendicular to it, and then third will be the equatorial region, where you will get the mirror images of both the sides, which we see it as bilateral cleavage. Then the third one was the spiral cleavage, which we had to discuss in detail about it. When we talk about the spiral cleavage here, generally the spiral cleavage plays an important role in mollusks. We see it where the cells are not arranged in a proper order. This is called as a holoblastic. I think it's not clear. I'm the shade because I'm taking it from the mobile. It is not so clear. Bear with me for today. And there is no support at home also to help me out in this. Fine. So it is a holoblastic cleavage where the new blastomeres are slightly displaced, giving a spiral appearance to the embryo. Spiral appearance to the embryo. It is just like, have you seen this small bubbles? Like nowadays we are getting those jellies. When we put it in water, they blow up. And those jellies, if you just put it small jellies, if you are putting it in water, they will not be arranged in a sequence order. They will just clump up, and they will be one above the other. They are not arranged in the proper order. Spiral cleavage appears to be same like that. So spiral cleavage here, they are not arranged in the proper order. They will be slightly displaced, like one above the other if we are keeping. And if it is not exactly on the above surface, it is slightly moved towards one side and the other cell is sitting in its place, how it will give an appearance that it is not an equal one but it is a displaced one. So in spiral cleavage, the blastomeres are spirally arranged. Example, it occurs in polyclads, nematines, annelids, mollusks, except cephalopods. In mollusks, except cephalopods, that is in octopus, etc. In all the others where shell forming is there you will see that the spiral cleavage is present. It is characterized by oblique orientation of spindles. What do you mean by this oblique orientation of spindles prior to cleavage? That means when the cleavage is occurring, the mitosis has to occur in every cell. And this mitosis is nothing but the cell nucleus that is there. It is opening up and removing the chromosomes, and these chromosomes are being pulled towards towards the corners in the anaphasic stage, and then the cytoplasmic division is occurring. This mitotic spindle, it should be on an equatorial plane, but it will not be in equatorial plane. It will be slightly tilted or it will be oblique. Now the cell that is dividing, it will be an oblique division. It will not be it will not be a perpendicular division, but it will be a slightly oblique division. I'll just show you that here in the figure. I don't have the cursor also. In the first figure, 4 cells what I have drawn. There, you can see the spindle orientation in different angles. The spindle that is present, that is small diamond shape what I have given, that spindle, it should be in the equatorial region. In the second one, you are seeing that it is oblique. One is towards one side, the other one is on the other side. As a result, the number of cells what you are getting is totally different. So in a spiral cleavage, you will see that they are slightly displaced one above the other, and the spindle orientation will be little different. The spiral oblique orientation of spindles causes the spiral arrangement of blastomeres. Spiral cleavage is a modification of radial cleavage. First 2 cleavage furrows are meridional. In radial and other meridional cleavage, and in spindle, it is perpendicular to the main axis of the egg. So what is happening in radial, you will see that the first is a meridional, the second one is a equatorial plane, and it is perpendicular to the first one. But here, in case of spiral, you will see that it is slightly inclined to the main axis. During the second cleavage, the 2 spindles are not only inclined to the main axis, but also they are opposite direction to each other. 2 spindles that are being formed in a cell, they will be opposite to each other. All the cells will not have the same type of the spindle formation. Therefore, the cells will not be equally divided. They will be divided in a different angles. As a result, the daughter blastomeres from each cleavage pair, they lie one above the other. That is one opposite to other. So in the same level only, but they will be instead of sitting in the same order, they will be placed in a different angle. So here these cells, now we name it as A, B, C, D cells. Now generally what happens, these cells A, B, C and D should be in an equal order. Now A is dividing in a horizontal plane, for example. B is dividing in a meridional plane. C is dividing in an oblique plane. Now what happens? The cells that are there, they will be dividing in different one, and the daughter cells that are there, they occupy a different place. So to identify the spiral cleavage, we will be naming them as ABCD cells. And these ABCD cells, they will be taking up a different position. Now the third cleavage that is there, it is a latitudinal cleavage. And these latitudinal cleavages, again, they are oblique to the main axis. They are not directly perpendicular, but they are in an oblique position. As a result, the animal pole, the micromeres and the macromeres that are there, they occupy different direction. They are not oriented to be placed on one above the other, but they will be displaced here and there. These spiral cleavages that are there, the third cleavage that is there, even this third cleavage, this will be more towards the animal pole, and it will be displacing the cells in a clockwise manner. As a result, what happens, you will see a dextral spiral cleavage. That means when it is moving in a clockwise direction, the shell formation or the cleavage that is there, it is towards the dextral side, that is towards the right hand side. Similarly, if these cells are oriented towards the left side, then you will see that they are going towards the sinistral side. That is how we get the different type of shells in case of mollusca, that dextral and sinistral cleavage, what we see, this is forming the dextral and sinistral torsion in case of molluscan. This is occurring just because of the cleavage that is not perpendicular or equatorial, but it is oblique to each one. As a result, the micromeres and the macromeres are not arranged properly one above the other, but they are being displaced little so that it gives a spiral image. So generally, the dextral spiral cleavages, they alternate with each other. The third cleavage that is there in the dextral, it is always alternates with the sinistral one. As a result, you will get even an odd number of cells. So even in odd number of cells, it will not give the equal number of cells. As a result, it leads into a cleavage which is completely spiral. And then the successive cleavages, the micromeres continue to divide equally, and the macromeres continue to divide unequally. As a result, all the cells are being displaced and they occupy a different position. And that is how we get the appearance of different cells. The spiral cleavage, each cell is distinguished, or it is completely distinct, and they develop into a particular part of an embryo, and it produces a mosaic type of a determinate cleavage. So you see that in this figure, you see the first one, the cleavage is occurring at different angles. As a result, the orientation of the cells are totally different. And you see that they are arranged in a different pattern, the macromeres and the micromeres, and the cleavage will occur in the micromeres further, and macromeres will be a slow cleavage that is occurring. As a result, they will be arranged just like a mulberry tree, the fruit. So in the spiral cleavage, you see that there is a disorientation of the cleaving cells because of the mitotic spindle, which are present obliquely, and the oblique divisions are taking place. The second type of division is the meroblastic type of division. In the meroblastic, you see that only a portion of the egg divide, only the because rest of the cell is completely filled up with the yolk. As a result, you see that only the nuclear portion, which is having a small amount of the cytoplasm, forms a blastodisc. And it is a partial or incomplete cleavage because only the upper portion of the cytoplasm that is there only will be dividing. And this is a characteristic of telolectic and the megalecytic eggs. That is, telolectic means large amount of egg yolk is present. Only a little amount of the cytoplasm and the nucleic position, that is the animal pole, is present. As a result, only a little amount of division will be occurring only in the anterior region. You can see that I have drawn two cells. Only in that particular portion, the division will be occurring. Then this meroblastic cleavage is of two types, discoidal cleavage and the superficial cleavage. Discoidal cleavage generally occurs in fishes, reptiles, and birds. Here, the cytoplasm is placed at the animal pole. It is in the form of a disc, and it forms a blastodisc, and only that disc will be dividing. Whereas in a superficial cleavage, it is characteristic to the centrolectal egg. That means the egg yolk is present in the center. So only the segmentation will occur on this superficial layer, and the egg does not extend, that the divisions will not extend into the yolk. This you will see it in insects. Then, based on the potentiality of the egg, the blastomeres that are there, how the blastomeres are formed, the cleavage can be divided into determinate cleavage and the indeterminate cleavage. Now, determinate cleavage determines what is the position of a particular type of a cell. So, cleavage producing predetermined blastomeres develop into a definite organ is called as a determinate cleavage. So it will form a dorsal end or a ventral end or animal pole or the vegetal pole or the animal anterior portion or the lower portion. So that kind of differentiation will be there in the cell. You can see in the diagram A, B and P1 what I have written. So dorsal side and the ventral side. Dorsal side, it will form anterior portion, posterior portion, and the ventral side, it will be forming the upper cell in the pole. When the division occurs, they will be going to the lower region and they are determined what it is going to form. So such embryo, which contains a different types of predetermined blastomeres, it gives a mosaic embryo and develops into a mosaic egg. And this egg will definitely have a spiral cleavage and gives a determinate development. It is in case of ascaris. Whereas Indeterminate cleavage is the blastomere. It is not determined whether what the cell is going to become in future. That is not determined. Generally, this will happen in case of all the other vertebrates, because here the development, when the cell has to become a limb, when the cell has to become an eye, it should have some kind of induction. There are certain organizers in our body which produces the chemical that you have to become this particular cell. It should be placed at a particular position, surrounded by a particular type of a cell. Then only it will develop into a particular organ. So they have done certain experiments where they have seen that this is called as a Spearman's experiment. What they have done is, during the developmental stages, when the morula is there, which is converting into a gastrula, during the gastrula, they know that the four layers are being formed. That is, three layered cell is being formed, which is giving rise to ectoderm, mesoderm, and the endoderm. From there, they picked up the cells which are determined to form a nerve cord, brain. That cell they have taken and then put into a tail of the developing embryo. At the tail region, because of the organizers and the inducers, because already it got that it has to become a nerve cell during the differentiation. So if you take that particular cell and keep it in the tail, second brain will be developing. So that means organizers and inducers play an important role in the development. So in case of vertebrates, all the cells that are developing during the developmental stages, until unless they are being differentiated into a particular cell, they all will be developing into a new embryo. They have the capacity or a potential to develop into a new embryo. Therefore, induction is required for those particular cells. Then that means that word is called as equipotential. What do you mean by equipotential? All the cells have the capacity to develop into a new individual. Every cell has the capacity to develop, whereas once a differentiation occurs during the gastrulation, you see that they are determined to become that particular type of cell. So that type of cleavage is called as an indeterminate type of cleavage. Then what are the factors that are affecting the cleavage? The rate and the nature of the cleavage is affected by factors like light, temperature, medium, amount of the yolk, cytoplasmic organization, mitosis, etc. The most important ones are the yolk, which influences the cleavage in two ways. One is the rate of the cleavage, as well as it determines the pattern of the cleavage. We have already seen large amount of yolk is there. Then the yolk cannot be divided into two equal parts. Easily, you cannot cut it into two parts. It is not easily acceptable for division. So division will be slow there. Whereas in the cytoplasm, division will be more. As a result, you will see that there are different types of cleavage. In the cytoplasm, cleavage will be more, and in the yolk region it will be less. As a result, you will get macromeres and micromeres. So yolk will tell you what is the nature of the cleavage that it has to occur, what type of blastomeres it has to form, micromeres or macromeres, equal type of macromeres and micromeres has to be formed, or it has to be unequal type of macromeres and micromeres, blastomeres has to be formed. It all depends upon the type of the yolk. So yolk plays an important role in the type of a cleavage. We have already seen in case of holoblastic cleavage and the meroblastic cleavage, holoblastic complete egg is dividing because there is only little amount of yolk is present. Whereas in meroblastic, large amount of yolk is present. As a result, it will be having very little amount of cleavage occurring. Only in the portion where cytoplasm and the nucleus is there, cleavage will be occurring. The second influence is made by the cytoplasm. Now, generally, the cytoplasm plays an important role in the cleavage, because here, during the mitosis, when we are talking about the mitotic phases, the cytoplasm, wherever the large amount of the cytoplasm is there, it is easily acceptable for the division. Whereas wherever the nucleus is present, the nuclear division, it will take larger time for the cleavage to occur. Though the cytoplasm will be dividing slowly, it will divide into two, but the number of cells that are been dividing in the cytoplasm, it will be more than the nuclear portion. motion. Then coming to the mitosis. Mitosis also plays an important role in cleavage in a sense that generally the cytoplasm is easily divisible, whereas the nuclear divisions take large time. The mitotic divisions has to take place so that the cytoplasmic division has to occur. In some cases, it so happened that the cytoplasmic division occur, but the nuclear division does not occur. And sometimes it so happens that without the nuclear division, cytoplasmic division can occur. That is how we get the polar bodies. During the formation of egg, we get the polar bodies where the nuclear division is not occurring, but the cytoplasmic division is occurring. So there are certain times that if the mitosis is not occurring properly, the cytoplasmic divisions can go on and the cellular divisions cannot take place properly. So mitosis also plays an important role in the cleavage pattern. Now coming to after the cleavage that is being done, we have seen that the cleavage is giving rise to the blastula. That is a single layer of large number of cells are being formed, and these large number of cells, because they cannot hold it tightly as a ball of cells, they try to arrange themselves in a single layer of cells having a small cavity, and that stage is called as a blastula. And during this formation, all the cells are equipotential. They are not determined to form a type of the cell. So next stage is it has to become arranged, furthermore arranged, and that process is called as gastrulation. That process is called as gastrulation. I've just directly taken down from the material, so it will be easy for you to understand the diagrams. You can see the diagrams here, the cellular region that is there, animal pole and the vegetal pole, and the formation of the cells that are there. When the blastula is converting into gastrular stage, you see that the cellular arrangement is different. It has to organize itself into a three-layered cell structure. And that three-layered cell structure, if you see the diagram here, vegetal pole, that is, other side is the animal pole, and the endoderm. Then you have a mesoderm towards one corner, notochord, neuroectoderm, and epidermal ectoderm. So ectoderm is outside, endoderm which has to go inside, and the mesoderm is flanking the endoderm, and the neuroectoderm and the notochord, it is present towards the ectoderm. In the next figure, when you see the blastocele that was more here, it is reduced. Endoderm is entering inside, it is pushing inside. You can see the fold, that black color precaudal plate that is there. It has invaginated inside. When it is invaginating, in the third one, you will see that there is a formation of a gap which is called as the archenteron. And that archenteron will be lined by the endoderm. And that endoderm will lead into the formation of endodermal organs like alimentary canal, etc. You can see this again. The blastopore that is converting into the archenteron, and the archenteron deepens, and the mesodermal cells will come towards the corner. And finally, you will see that archenteron is there, notochord on the top, and the mesoderm towards the middle region, and the endoderm, it is flanking the archenteron, and the dorsal lip and ventral lip are the lips that is from where the folding of cells are occurring. So this is just to imagine how the plug or as the archenteron is being formed. Let us see what exactly you mean by gastrulation. Gastrulation, it is a development of gastrula from blastula, wherein the single layer of the cells will be converting into three layers of cell. During gastrulation, the blastomeres that are arranged in three layers, they are the ectoderm, endoderm, and mesoderm. Now, these cells that are moving inside, when they are moving inside and forming a three layers, this phenomena is called as a morphogenetic movement. This morphogenetic movement, they give a definite shape to the embryo. Hence, the name it is called as the morphogenetic movement or the embolic movements. Embolic movement. And this two movement, that embolic movement and the epibolic movements, these are the two movements of the arrangement of the cells which will give a different type of structure to the cell. We will come to it little later, like what are epibolic and what are the embolic movements. Now, before that, we should know what are the events that are taking place during the gastrulation. There will be a groove appearing below the gray crescent. Now, what is this gray crescent? Generally, during the blastula converting into a gastrula stage, what is happening during this particular phase is that the cytoplasmic differentiation is occurring. What is that cytoplasmic differentiation? Like, for example, you see a mango. When it is very small mango, when the seed is not being formed, the outer shell, that is outer skin, inner mass, and the seed that is forming, it is also of the same nature. Slowly you will see that a small layer is being formed. Then you will see that, okay, this is a seed forming area. So like that, you will have certain differentiation. Similarly, when you are seeing the blastula, all the blastomeres are of same size. The cytoplasm that is present, that cytoplasm gets differentiated. That I have to become an ectoderm, I have to become a mesoderm, I have to become an endoderm. Like that, they will be forming. The cytoplasmic differentiation will be appearing. And when this is appearing, they will be just marking, having the cytoplasmic coloration will be different, which is visible onto the embryo. With that, we will be able to mark out that these are the differentiation layers of the cells that are there. And the notocaudal cells that are there, notocaudal cells, what do you mean by the cells which are forming the vertebral column? They are the notocaudal cells and the nervous system. Those cells, they will form a darker color and they will form a small crescent-like structure, and that is called as a gray crescent. So the groove appears below the gray crescent, and the macromeres move into the embryo through the groove to form an endoderm. That is what I have shown you here. The macromeres will be moving inside to form the endoderm. And this movement is called as invagination. So invagination will be occurring. When the invagination is occurring, the archenteron is being formed. At that time, there is an opening. And this opening that is being formed, that is called as the blastopore. And this blastopore will be guarded by two lips, that is a dorsal lip and a ventral lip. This endoderms will be moving inside, and once they are moving inside, the yolk plug will be formed. That means that further development or the further thing will not be forming, and they will move towards and closing that particular area, forming the lips, and that is called as the yolk plug. And after that, the mesodermal cells, the precaudal cells, the notocaudal cells, they all will be migrating through the blastoporar lips, and then they will converge to a particular position, and this movement is called as convergence. And after that, the ectodermal cells that are there, they will roll over everything because they have to cover up the entire cell. So that is called as an involution. They will be projecting out, and then they will be covering up the entire cell. And this projection, this is called as a divergence movement. And the precaudal cells, the notocaudal cells, they will go and arrange themselves in the mid-dorsal plate so that they have to form the nerve cord and the notochord. Now, then once this mesoderm, you can see in this particular area, the neural plate formation of cells, the notocaudal cells, the archenteron cells, the blastopore, the yolk and the mesodermal cells, then epidermal cells towards the outside, and the endodermal cells towards the inner. This is a gastrula that is being formed. In general, what we write is a 3 layers: ectoderm outside, endoderm inside, and mesoderm in between that. But during the morphogenetic movements, you will see that these are the changes that are occurring. So this is what is being said. So then as the cells are moving, you will see that the gastrula is becoming mature or oval in shape, and the archenteron, it will increase and it will lead into the blastopore. And the cells start differentiating into outer ectoderm, inner endoderm, middle mesoderm, and the ectoderm again will be divided into notoectoderm and a neuroectoderm. So the gastrula now can be called as, it is the embryo which is developed from the blastula. It is oval in shape, which contains 3 germ layers. They are the ectoderm, endoderm, and mesoderm, and it has a cavity called as an archenteron, and it opens to the exterior by means of a blastopore, and the endoderm cells protrudes through the blastopore, and these cells constitute the yolk plug. And the floor of the lateral sides of the archenteron are flanked by the endoderm. A roof of the archenteron is by the cauda mesoderm, and externally the gastrula is covered by the ectoderm. So these are the layers. Now let us see how these morphogenetic movements are occurring. So when the cells are moving inside, what exactly the movement is taking place. These movements, they are called as epibolic movements and embolic movements. Now, epibolic movements, it refers to the movement of the cells on the surface of the embryo, the ectodermal cells that are there, that is the epidermal cells, the epidermal cells, like which is forming the caudal cells as well as the neural cells, they will be entering inside, they will be invaginating and going inside. So first process is invaginating. So the ectoderms and the ectoderm that is there, it will be epibolic, it will cover up the area, so it will move outside, that is called as involution, rolling of cells outside. So the cells, the cauda neural cells will be entering inside, and they spread out antero-posteriorly in direction, and they extend in as a narrow strip, and they go and settle down in the roof of the gastrula stage. So epibolic movements are those movements wherein you will see that the cauda neural cells are entering inside. I will show you the last figure, then you will understand this. So the cells, what we have seen there, the notocaudal and neural cells, they will go inside. The mesodermal cells which are there, they will come and flank towards the corners. And the endodermal cells, they will go and bind, or they will cover up the archenteron, and it will form the endodermal organs. So that is how the cells will be arranged together, the mesodermal cells towards this corner. So in this, what happens during the development, the mesodermal cells that are there, they will form the muscles, the bones, etc. The endodermal cells that are there, these endodermal cells, they will develop into the internal organs like alimentary canal, or the kidneys, the heart, the lungs, etc. All these endodermal organs, they will be forming. And the ectoderm that is there, the outer lining, it will form the skin covering the ectodermal layers, and the notochord will form the vertebral column, and the neural cells, they will be forming the nerve cord. This is how the ultimately the arrangement of the cells will be there. So now, this morphogenetic movements we were talking about, the first was the epibolic movement, wherein the movement of the surface of the cells will be taking place on the surface. The cauda neural cells will be entering, and the epidermal cells try to cover up this. And during this covering up process, the two lips are being formed. That is, the ectodermal lips are being formed, dorsal lip and the ventral lip. Then emboli, it refers to the movement of the cells from the surface to the interior of the embryo. So presumptive endoderm, mesoderm, notocaudal cells, they will be migrating inside, and they have the space called as the archenteron with it. And this embolic movements are of different types. They are convergence, involution, divergence, invagination, polyinvagination, ingression, delamination, concrescence, and the last one, now, that is infiltration, constriction, extension. All these are different movements of the cells wherein they will be covering it up and taking up their spaces. Now, coming to convergence, it refers to the movement of the cells toward a single point. In chick and mammal, the cells from the surface of the blastomere move towards the primitive streak formation. What is this primitive streak? Primitive streak is the arrangement of the cells along that axis. The development of the neural tube and the myotomes will be seen. So primitive streak formation is an indication of the developing embryo. Whereas in involution, it refers to the rolling of the cells. They are located on the blastoporal lips. I told you that involution, what is happening? The ectodermal cells that are there, they will be just rolling up from covering up that particular pore. So during that rolling up, they will form a lip, that is neither they are completely inside nor they are going outside. So they roll over each other and forming a lip. And that particular position, it is called as involution. It refers to rolling of the cell. The cells located on the blastoporal lip move over the lip, and they brought that means they overlap each other, so that they completely form, or they will what do you say, when the cells are covering it up, the primitive streak will have the capacity to develop further, and these cells will be covering up the primitive streak, and they will put the primitive streak in a particular groove. They form a groove or the invagination for the primitive streak to form. So the cells which are rolling inside and covering up the primitive streak that are there, the cells that are arranged, and the covering up to it, they form the groove for the primitive streak, and that movement of the cells is called as involution. Divergence. Here the cells move in a different directions from a single point. The migration of involuted cells from the blastopore to, or the primitive streak to the interior of the embryo. This is called as the divergence. They are moving to the different positions. They are moving away from the primitive streak to hold on the different positions. Invagination, it refers to the infolding or inpushing of the layers of the cells. This process will lead into the formation of the ectodermal cells, like the cauda ectodermal cells, the neuroectodermal cells, and the endodermal cells. So invagination will occur initially with the ectodermal cells to form the cauda neural cells. Polyinvagination. Now all these phenomena, one more thing, all these phenomena will not occur in only one particular cell, in different, like epibolic movement. Convergence will occur in some cells, involution occurs in some cells, divergence will occur in some. Like example, invagination, we are seeing that in case of amphioxus, this process will occur. In polyinvagination will occur in case of amniotes, like for example, chick, where the amnion layer is there. So number of invaginations will be occurring. So here the outer layer invaginates many times at many places, because the amnion layer is there. This amnion layer has to be moved inside. So there it will occur many times. So it is not that each step will be occurring in a particular egg. Like for example, we are taking a chick egg, it is the embolic movement, it will be in a polyinvagination form, which will be leading into the formation of different cells and the spreading of cells. That is, the delamination will occur, and then they will be arranging properly. So like one of the movement will be occurring. Ingression. It occurs in the embryos of reptiles, birds, and mammals. During this phenomena, the individual or the group of cells separate from the primitive streak and migrates between the epiblast and the hypoblast. This epiblast and the hypoblast, these are nothing but the area wherein the like you will see the formation of the cytoplasmic differentiation, which is going to form the outer layer, and the hypoblast is the middle layer and the endodermal layer. That is, during the fate max, you will see that the cytoplasm is divided into the epiblast, endoplast, and the hypoplast layers. So it will be moving towards the outer region or towards the posterior region to form the cauda neural cells or the endodermal cells. Delamination, it refers to the separation of the layer of cells from another layer by splitting. This separation will occur from endodermal and the mesodermal cells. So now what happens is, when the cells are being arranged, during the fate map, I told you that in the fate map, the cytoplasm is totally differentiated. So these cells, they will occupy a particular place. Now, once they are occupying a particular place, say for example, when we take a ball of cells, the upper layer of cells, they are epidermal cells. The below layer of cells in a ball, they form the endoderm. And the side layers, they will form the mesoderm. Now, mesoderm, endoderm has to go inside, and the ectodermal cells have to come outside. So what is happening during this process? First, the ectodermal cells that are there, these ectodermal cells, they should roll outside to cover the endodermal cells, etc. So they are rolling, epibolic movements are taking place, but the notocaudal cells that are there on the epidermal layer, that has to come inside. So when they have to come, so they will be embolic movement of the cauda neural cells. Did you imagine now? You just imagine one ball of cells, one single round of cells, wherein our upper layer is ectoderm, lower layer of cells are the endoderm. Towards the sides of that ball, it is the mesoderm. The mesoderm, endoderm has to come inside. ectoderm has to cover up all the cells. So the ectodermal cells, they will be moving outside. They are the epibolic movement. And the cauda-neural cells that are there, which is going to become the nervous system, and the caudal cells, they have to go inside. So they will be going in deeper inside. So that is involution. Emboli will be taking place. And the cells that are to form the epidermis, they will be moving outside. And that is the epibolic movement. So they will be moving outside, covering, and thereby dorsal lips are being formed. So now these cells, when they're moving inside, they have to arrange itself. So from single layer, they have to form a particular cell, they have to spread out into different layers. Now, the inner layer, you have seen that there is the endodermal cells and also the mesodermal cells. Now, endodermal cells, they have to ingress, they have to be going deeper inside the cellular structure, having the cavity called as the archenteron. So they will be ingressing inside. So they will be moving inside, invaginating and inner. So they have to separate from the mesodermal cell. That process of separating from different layers of the cells is called as delamination. Then concretion, it refers to the migration of the group of cells towards each other until they meet and fuse together. This will occur during the development of feathers in the birds. So this congregance, that is the two cell, that is when the opening is there, that opening has to be closed. So when both the cells will try to meet together, that particular phenomenon is called as congregance. Then infiltration. Infiltration is nothing but it occurs during the development of endoderms in amniotes. Individual cells detach themselves from the surface of the blastoderm and fall into the primary blastocele. Here they arrange themselves in a single layer of endoderm. So infiltration is nothing but it is a movement of the cells separating from the blastodermal layer and forming a different type of cell. Then constriction. It refers to the reduction in the size of the blastopore. The two lips that are there, they have to close the blastopore because the blastopore also plays an important role in the development. That is, either it will become an anus or it becomes a mouth. If it is becoming a mouth, it is that is it. They are the proteostomes, and if it is becoming the anus, then the mouth opening is there, the deuterostomes. So like that, this blastopore opening and closing plays an important role. So the constriction should occur here, whether it is developing into a anal region or the alimentary continues with the alimentary canal, or it is closing and forming a mouth. So depends upon, and that is how we get the deuterostomes or proteostomes. And elongation, and that is extension or elongation. It refers to the antero-posterior elongation of the part of the embryo. Now the embryo, till now it was a ball of cells or it has become oval. Now it has to elongate so that the complete structure of the embryo primitive streak also elongates, and the development will take in the antero-posterior axis, because the development occurs in the antero-posterior axis, and some of the development will occur in an oblique axis. Some of them will occur at the equatorial axis. So what are those cells which are undergoing the body plan? When we talk about the body plan, like for example, if I talk about hands, hands cannot develop in a vertical axis, isn't it? They have to develop in a different axis. Similarly, all different types of organs that are there, they have a different plane of their development. So based on that, the development, you'll see that the body axis plays an important role. When the body axis is playing an important role, you see that the development of the animal will take place in a different axis plan. So there is a different axis for the neural development, for the alimentary canal development, for the development of the limb buds, for the development of the other vital organ. There is a body plan or body axis, and based on that body axis, you will see that the development of an individual occurs. So to summarize the gastrulation, bastula and gastrulation, the morula dividing into number of cells by means of cleavage, depending upon the nature of the yolk, depending upon the cytoplasmic constraints, depending upon the mitosis, the cleavage will occur in a different pattern. Depending upon the nature of the yolk, cytoplasm, they will give rise to the blastomeres, and the blastomeres can be micromeres and macromeres, and this ball of cells will be arranging the cells in a single layer, having a cavity called as a blastocele, and this blastocele, blastula stage, will have the differentiation of the cytoplasm, marking the fate maps onto the structure or fate maps onto the cellular structures, and these fate maps leads into the formation of the gastrula, where the differentiation of the cell will be occurring. They are being demarked to form the ectoderm, endoderm, cauda cells, and the mesodermal cells, and by the process of morphogenetic movements, it will form into a three-layered structure that is called as a gastrula. During this process, the epibolic and embolic movements will be occurring, wherein the cells will roll out or roll inside, having the invaginations and opening the archenteron, and then leading to the formation of the anal and the mouth opening, formation of proteostomes and deuterostomes, depending upon the blastoporal opening or the closing of blastopore. And they arrange by means of the epibolic and embolic movements. Due to invagination, the cauda cells will enter inside, and by the divergence or the delamination, you will see that the mesodermal cells and the endodermal cells separate out, holding the archenteron, and then they lead into the formation of endodermal organs. Whereas by the epibolic movement, we see that the epibolic movements and the involutive movements, we see the epidermis or the ectodermal cells will be rolling outside the embryo. It will be rolling outside the embryo and then covering up the entire embryo, making it a appearance of three-layered gastrula. Hello?
Yes,
Hello?
ma'am.
Yeah. Then tomorrow, like I have like group exam invigilation. Is this link open after 5 o'clock?
I don't know, ma'am.
After 5 o'clock, is this link? Because I have the exam till 5 o'clock. Group exams are there. I'm chief superintendent there. So may not be able to take class by 4, but I'll try to link in. Just at 3:30 or so, you just get in touch with the link if it is open, because there are jammers will be kept in our college. So
Okay.
if I'm not able to take it up, then I will cover up the entire portion in the next coming week. Okay,
Oh,
in the 15th and 16th,
okay,
I will
ma'am.
cover up the entire syllabus, taking extra hour. But tomorrow, you just try to log in at 3:30. I will try to join. At that time, I'll continue taking the remaining part. I'll try to complete the embryology part. Differentiation and limb development, I will complete it off tomorrow.
Okay, ma'am.
Okay, if not, you just try to see if the link is open after 5. Today, you try to see whether the link is open after 5 o'clock. If it is there, you just convey it on the WhatsApp group, or one of you have my number, no? You can just convey it to me so that 5 o'clock I'll join you.
Okay,
Okay?
ma'am.
Okay, ma. Thank you.
Hi, ma'am. Thank you, ma'am.
Okay, ma. Bye.